Welcome back, science fans. Today we're going to be talking about the process of mitosis, which is how cells go about dividing. Now, there are three main reasons that cells will divide. The first is that they want to grow larger, or organisms need to grow larger. The second is so they can repair dead or worn out cells. And the third is so they can reproduce. Let's look at each one in more detail here. Alright, the first reason that a cell needs to divide is so that an organism can grow larger. One strategy for organisms to grow is that they split into multiple cells, and so we end up with multicellular creatures like you, me, hamsters, turtles, all things like that. But there are single-cell organisms out there like amoebas. So why are we not single-cell creatures? Well, the reason is this. As a sphere, okay, which is sort of the shape of a cell, gets larger and larger, the ratio between the surface area and the volume decreases, which means that the outside does not increase as fast as the inside. And this creates a problem for a cell because remember, around the outside of a cell is the cell membrane, and its job is to bring nutrients and things that the cell needs into the cell. And because the surface area isn't increasing at the same rate as the volume, so it's not getting as big as fast as the inside, it can't keep up with the nutrient demands of the inside. Plus, there's a further distance to transport those nutrients to the center of the cell. So it's a further distance. So rather than just keep getting bigger and bigger, it makes more sense to divide and have multiple cells with a smaller surface area to volume ratio, or a larger, surface area to volume ratio. All right, that's growth. All right, the next reason that cells divide is so that they can repair or replace dead or damaged, worn out cells. Now, over time, this happens in multicellular organisms where you have many cells. Cells will die off and we need to replace them with new cells. And so that's repair. The next reason is that we'll get cuts, we'll get scrapes, and we'll have damaged cells. And again, we need to replace those, which is a good thing so that we don't have all these cuts and scrapes all over us that we've collected over the years. As well, we're constantly replacing cells just as they become worn out. You'll have skin cells flake off over time, kind of gross, but that's what's going on. We're replacing those cells with new cells, and that happens other places in your body as well. You're constantly making new blood, which is why you're able to donate blood and you don't run out, okay? Because your body regenerates new blood cells when some are removed, and that's repair. Another reason that cells will divide is that so they can reproduce. Certain organisms are made up of only one cell, and for them, they reproduce asexually, which means they don't need a mate to reproduce. So that single cell, what it'll do is it'll split itself in half and become two organisms, and therefore reproducing. So for them, the strategy to reproduce and increase their population is to split in half, and therefore the cell needs to divide. So it's reproduction. So, now that we know why cells divide, how do they do it? What's the big challenge? Well, the big problem is that a cell has DNA. It's the genetic information, which is really the blueprint or the plan on how to go about and make the organism. Let's say this is your DNA. If you started off as a single cell, if that cell split its DNA in half, and then each cell got half of that information, it would only have part of the information on how to make you. And then the next time those cells split, we'd only have less and less and less. Every time the cell divides, there'd be a smaller piece of the information. And since you're made up of trillions of cells, those cells have divided many, 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 many times. And you would be dealing with such a small piece of the blueprint that there'd be no way to make the organism. So how do we go about splitting up that DNA? So let's introduce you to some terminology. The starting cell that we're looking at that's going to divide, we call that the mother cell. The cells that are produced, when it splits in half into two, are what we call daughter cells. Okay, so we have a mother cell producing two daughter cells. Now, we know that each daughter cell has to get a full set of the DNA. How does that happen? Well, obviously we need to make a copy of the DNA before we divide. The same is true, we need to make sure that each daughter cell gets organelles, it needs mitochondria, it needs endoplasmic reticulums, those things so that it can go through and survive. Now, those parts are important, each one needs to a set so it can do that. How does that happen? Well, it happens through the process of mitosis. Let's go through the different steps. All right, let's take a break here for a second, and I've just got something to share. I wanted you to know that I poop mostly after tacos. Maybe a little personal? Well, here, let's take a look. I poop mostly after tacos. If we change that, we'll see that IPMAT, interphase, prophase, metaphase, anaphase, telophase. Those are the phases of the process of mitosis, which is the name we give to is how a cell goes through and divides. So, IPMAT, I poop mostly after tacos, interphase, prophase, metaphase, anaphase, telophase. That's what we're going to use to help us to remember the order of the phases of mitosis. Let's look at what's going on in each of these phases in detail. Now, it's not uncommon for cells to divide every 24 hours. Most of that time is spent in interphase. Very little is actually spent in mitosis, as we can see here in the cell cycle. So what's going on there? Well, the cell needs to rest a little bit after it splits. It needs to grow, get larger. It needs to copy its DNA. It needs to make sure there's enough organelles so each daughter cell can get a full set. And that's what's going on in interphase, where the cell spends most of its life. Now, the first real phase of mitosis is called prophase. In prophase, several key things happen. First off, the chromatin, which is the genetic information, is going to shorten, thicken, and form into what we call chromosomes. So that's the genetic information. Now you'll notice each chromosome sort of has this shape here. It is two sets of your DNA, and each chromosome is bonded together at the middle with what we call a centromere. Each half has the exact same genes. So there might be a gene for eye color on one half of the chromosome, and on the other half, which we call chromatids, have the exact same information. Okay? Now, another thing that happens in prophase is that the nuclear membrane, the membrane that surrounds the nucleus, starts to disappear, because what we need is we need to get that genetic information out of the nucleus. Another thing that happens is the centrioles, they start to move towards the poles, or each side of the cell, and they start to form spindle fibers, which are going to be pretty important in organizing the chromosomes and how they divide. Okay, so that's what's going on in prophase. The nuclear membrane starts to disappear, the chromatins shorten and thicken into chromosomes, and the centrioles start to send out their spindle fibers and they move to the poles. All right, that's prophase. All right, metaphase. Now, back in elementary school, when you finished up recess, your teachers made you line up in long lines and just to keep things organized. The same thing kind of goes on in metaphase. What goes on here is that the chromosomes line up down the middle of the cell, and these spindle fibers that come from the centrioles, they attach to the centromere, which is the thing that holds each half of the chromosome together. So that's what's going on. They're sort of organized and they're getting ready. So metaphase, line up down the middle, centromere, or spindle fibers attached to the centromere, and we're getting ready for our next phase. That's metaphase. The next phase is anaphase. Now we know the big challenge, and the whole reason we're doing this is so that each daughter cell can get a full set of DNA. So we've got all the DNA kind of lined up down in the middle. What happens here is those centrioles start to tug on those ropes, or those spindle fibers, and they pull real hard until eventually the centromere, the thing at this middle of the chromosome, breaks, and we pull each side a set of DNA. And that's really what's going on in anaphase. We break the centromere, and each half of the cell starts to get a full set of DNA if everything goes correctly. That's anaphase. All right, the next phase is telophase. And in telophase, that genetic information that's been pulled to either side of the cell starts to have a new nucleus form around it by a new nuclear membrane appearing. Those chromatids, which remember is the name for half of a chromosome, which is a chromatid got pulled to each side, starts to unwind, okay, back to the way it was in interphase, which we call chromatin, and we really can't see it with our microscopes. So invisible chromatin now, becomes or is formed from the chromatids, and as well we get the nucleus forming around. That's really what's going on in telophase. Now often we lump in another process here in telophase, and this is a process called cytokinesis. And what happens is our cell membrane starts to now that we have the genetic information either side, start to pinch off, and we get our two new daughter cells that are smaller than the original, but are identical in terms of their genetic information. And now each of those daughter cells will now enter into interphase, so the cycle continues itself. And so each of those cells will start to grow, prepare, and copy its genetic information, produce more organelles, and it'll divide again. And that's what's going on over and over again inside your body and in all multicellular creatures. This is the process of mitosis. All right, so we've seen our different diagrams of what these stages of mitosis look like. Let's see what this really looks like under a microscope, and watch to see if you can see the different phases of mitosis that we've talked about.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
All right, so you can see that the actual process isn't quite as pretty as our little diagrams. Another thing to note, humans have 46 chromosomes. In our diagrams, we often don't draw 46. We'll draw, 2 or 4, maybe 6 chromosomes. And that's just to simplify what's really going on. When you see those diagrams, don't get confused. We actually have 46 chromosomes. All 46 would line up in metaphase, say, and split in anaphase. In our diagrams, we only draw a few to keep things simple, and we understand really what's going on. All right, science students, that's mitosis, and we'll see you back next time.